All right. How's everybody doing? Good. I hope you all had a great weekend. Today we are going to finish talking about some details of antibody function that I didn't get a chance to finish up last time. And then we're going to move into our discussion of the generation of antibody diversity. If you look at the slides that I've posted, this set of slides will be for both today and for Wednesday, instead of me having to try to figure out where to split things and details like that. So just know that this PowerPoint is for both days. We left off last time thinking about the different antibody isotypes and specifically what types of functions were associated with each of them. We saw IgM and its unique features and functions. We saw IgD and we saw IgE. We finished up at the end last time with IgA. And as we discussed about IgA, the biggest thing that is important about IgA is its anatomic location, more so than any other specific details of it. And so with IgA, we tend to see IgA, which is shown here in yellow, in mucosal sites. So you can see that it's in the respiratory tract, the GI tract, eyes, nose, mouth, all of those types of locations. And so remember that IgA is able to cross epithelial cell barriers to go into these mucosal sites and be able to protect you sort of even before potential infection, really prevent that microbe from establishing by blocking in the mucosal sites, while many of our other isotypes are really found kind of throughout the body in the blood, but not crossing into those other mucosal sites. I also mentioned that IgA is found in breast milk. And so if a mom breastfeeds the baby gets IgA from the mom in their GI tract. And so you can see that happening here. There was one last piece of information about IgA that is really mostly because people find it interesting, that I didn't have time to mention last time. So there is something known as selective IgA deficiency. This is the most common immunodeficiency. So of all of the parts of the immune system that might be missing, this one actually happens the most frequently. On the left is a table from your textbook giving the number of people who have IgA deficiency per million individuals. And you can see that this is broken down amongst different populations. And so this is per million. It's pretty high. And from all that we know, individuals are pretty much entirely healthy. So you could be walking around right now with IgA deficiency, meaning that you just don't have any IgA, and be seemingly healthy. Part of the reason for that may be due to modern hygiene, that we may not be encountering some of the microbes that past humans did. And so maybe in the past, people with this deficiency had more issues, and now because of sanitation, we don't have that. There are sort of ideas that perhaps people with selective IgA deficiency may have differences in maybe having some chronic lung disease, possibly due to air quality, or there may be an association with celiac disease, though neither of those are super clear epidemiologically. And one of the things that I think is particularly interesting here is that when we think about different immunodeficiencies, we can think a little bit about the fact that, well, some of them, may have been selected evolutionarily, negatively selected in the population. So if people were born with those deficiencies, they maybe got infectious diseases more often and died and didn't have progeny. And this one's kind of common. So it seems like there really wasn't that kind of selection against the disease in the same way that you might expect selection against against more severe immunodeficiencies. You might say, this is weird. Why is it that in the past, all the people with this selective IgA deficiency didn't all die and not have progeny, and thus, make it so we didn't have this anymore? And one of the things that we think is really important here is that with selective IgA deficiency, you would still get your mom's IgA. So even if you yourself could not make IgA, when you were an infant, when you were a baby, you would be still getting IgA from your mom. So during that period of time when you might be especially susceptible as a baby, you actually have IgA because it's from your mom coming through breastfeeding. And so it's possible that that has really impacted on the selection of this deficiency. So that's just kind of like the fun fact about IgA. The final antibody isotype that we need to talk about is IgG. As you can see here, there are multiple different subclasses of IgG. So here the textbook is showing you IgG 1, 2, 3, and 4. There are even more than that, but we're just going to go with this textbook. And if you look at them, you can kind of see places where IgG might be sort of standing out compared to some of the other isotypes. So if you look at these data, where might you notice IgG being sort of unique? And it's tricky because there's 4 of them, and they don't all give you sort of a perfect answer here. But yeah, Grace. So it has a much longer half-life in serum. And that sort of is related to the other thing that is sort of unique about it. Especially if you looked at, say, G1. Yeah, Emma. It's the highest serum concentration. Of all of the types of antibodies in the serum, IgG is by far the most. So if you were to take some serum right now and find some antibodies, most likely those antibodies would be IgG. And so this is the one that we know sort of a fair amount about. In a lot of experiments or a lot of things where we try to measure antibodies, we often are doing IgG measurements because there's just so much of it. Like we're not going to do IgE measurements in the serum as easily because there's so little of it. But usually if we're measuring a lot of antibodies in the serum, we are measuring IgG just because there's a lot of it. You can also see that IgG has some unique areas in terms of function. All of the types of IgG are really good at neutralizing. There's a little bit of variation with some of the others in terms of how well they, say, activate complement or do other types of things. IgG is really the only one that's involved with NK cell activation. It is pretty good with opsonizing, all stars at neutralization. So it can do a fair number of functions. It's pretty broad function-wise. You also saw that there are different subtypes listed previously, and here you can actually see the images showing the differences between those subtypes. And what you can notice is that the big difference in those subtypes is how big the hinge region is. So how much hinge region or how many amino acids there are in the hinge region. And so this table from the old version of your textbook gives differences in lengths of the hinge region. It also tells you about the frequency. So you can see most IgG is IgG1, then the next most is IgG2. The IgG3 has the shortest half-life. It also has the super long hinge, and that hinge can get broken easily. That's what keeps its half-life shorter. The other ones have shorter hinges, so they have longer half-lives. They can activate complement somewhat differently. They're susceptible to proteolytic cleavage differently, but in a lot of ways, they're decently similar, at least for our purposes at this point. Again, there are two big things I think about when I think about IgG. One of them has to do with the timing that it is produced during a response. I told you last time that IgM tends to be made early. I told you that the mnemonic of MDs give everyone apples also has something to do with an order of production. And so the point of telling you that was that IgM was made early. And so here you can see IgM in black, you can see IgG in red. Here we're getting sort of a first dose, maybe a first vaccine dose. Here we're getting a second vaccine dose. And so what I want you to notice is that early on, after that first vaccine dose, the antibodies are all IgM. So IgM is kind of the thing that tells you, like, you're making an early part of the response. If you drew someone's blood and saw they had a lot of IgM in response to something, you might know, oh my gosh, they're early on. IgG becomes the really predominant response later on. And in particular, the second time you are exposed to some microbe, you make way more IgG than you do IgM. And so in a lot of cases, people will be doing antibody typing to determine kind of how early or late in a response you are, or if you're in a secondary response, the second time responding to something, by looking for IgG. This will come up later on. So I'm going to tell it to you and you're going to be like, yeah, whatever. But later, this matters. In reality, you could actually measure IgG, IgA, or IgE to tell you about the same phase of the response. All three of those are kind of made in the same lateness of the response. So you could do A, G, or E at this point. But there's so much G. There's tons and tons and tons of IgG. Why do the hard one? When there's so much IgG, just do the easy one. So we measured IgG for the secondary response, both because it's good for that timing, but also because there's a lot of it, so it's easy to measure. The other thing that is interesting about IgG is that IgG can have a unique anatomic location as well. So in most individuals, we think of IgG as just sort of generally being throughout the blood, throughout the body, everywhere. So you see everything's pink. However, the one other unique situation is that IgG is the only type of antibody that can cross the placenta. The placenta is actually a great barrier that can stop a lot of microbes, a lot of things from going into the baby. In order to cross the placenta, there has to be a whole special process. And IgG has the ability to do that process and cross into the placenta. And so a fetus and a newborn will have a whole bunch of IgG coming from its mom. And just like in the case of IgA, this involves the antibody actually having to get carried across a layer of cells. So there's this nice cell barrier. In the case of the placenta, it's the syncytial trophoblast, which separates mom's blood from baby's blood. And we have this nice little carrier receptor that helps move antibodies across that cell layer. So IgG can cross the placenta, just like IgA was crossing epithelial cell layers into mucosal sites, through a relatively similar process. If we look at babies both before birth and then afterwards, you can see birth is here. We've got all the way out to -9 months. So this is, before birth, and then we've got months and then years as an adult. You can see at birth there's some newly synthesized IgM. There's not a lot of IgG or IgA being made until a little bit later. You can see in some cases out to like a year. But really, at the beginning, the first, say, 3 to 6 months, that baby's not making many antibodies of its own. In fact, the only antibody that baby really has is IgG coming from mom, that came from mom through the placenta, and IgA that may be coming if the mom is breastfeeding. Otherwise, that baby doesn't really have much of anything until about 6 to 9 months when antibody production really starts in. This has a lot of really interesting implications and a lot of things that can be kind of cool about it. One of them is that this actually goes into a conversation I sometimes have with regard to science communication stuff. It's one of those arguments I have to argue with people on the internet about a lot. There are a bunch of people online who get really angry about childhood vaccinations. And they say, I cannot believe we give babies so many shots all at once. That's too many shots all at once for them. Right now, standing under this air vent, I am being exposed to more microbes than are in those shots all at once. So not really, their immune system can take it, just like my immune system can take standing under the air vent. But the reasoning why we do those vaccines at specific times is because we're basically vaccinating at the first time where the baby's immune system is able to do something. If we went before this, the baby's immune system wouldn't be ready yet. We could wait longer, but the longer we wait, the more time that baby's sitting around being susceptible and possibly getting sick and dying. And so basically we know this information, we know where antibody production is going to start. We know that's also in a lot of cases about when mom's antibodies go away, and we say, we got to protect you right now. We're going to put those vaccinations right at this point in time. So these types of data are used in doing things like designing the vaccine schedule, and are also really interesting in some other ways. There are some infectious diseases, the most famous of which is pertussis, which is extremely deadly for infants. Pertussis, like if you get pertussis when you're, 10 weeks old, it's kind of lethal. Pertussis is really, 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 really bad for babies. You also will have heard of whooping cough. Pertussis is whooping cough. So if you look at these sorts of data, you can realize one potential problem that we as immunologists might have in trying to protect babies against pertussis. Why would it be really hard to protect little babies from pertussis based on these data? Yeah, Michaela. Yeah, you basically can't vaccinate the baby that early to get them the immune responses that you need. As a result, there has become a new strategy. And this strategy, I would say, has really taken off in the past 10 years or so, which is that now we give extra doses of the pertussis vaccine to pregnant women with the idea that we make the mom make a bunch of antibodies and give the antibodies through the placenta so that the baby gets antibodies from mom coming through the placenta. Some of you guys are making great faces. That's just so cool. Yeah. And so in fact, I have a friend who does a lot of work with what's called maternal fetal immunology, and she's actually her whole lab is about how to do different types of vaccines in moms to protect babies. All to try to basically take advantage of the fact that IgG can go through the placenta. And so now, in fact, within the past 10 years, the FDA has added a recommendation for women in their third trimester to get a pertussis vaccine, specifically to protect the baby early on. So this is the kind of thing you can get from knowing these sort of basic pieces of information. One sort of downside of all of this, just to know, is that if mom has, say, a disease that is mediated by antibodies, like an autoimmune disease that happens because of antibodies. So here, mom has Graves' disease, and in the Graves' disease, in this particular case, happens because of antibodies to the thyroid, all dramatically not to scale. It does actually also have some eye bulging symptoms, but like I find this picture to be very odd with the scale of all the things. In any case, if the antibodies are actually the thing causing the disease, the antibodies can cross the placenta and cause harm to the baby. And so you can see situations where, like in this case, this newborn might suffer from the same antibody-mediated disease. However, we have a lot of good ways to actually deal with that now. Now, if we know mom has an antibody-mediated disease, we can sort of deal with this. We can give some treatments to the baby. There's one other thing to point out about this. So this is listed as passive antibody transfer. Basically, if you're just, and because the idea is we're just transferring antibodies. We saw passive antibody transfer when I was talking about treating black widow spider bites and all of those other things with antibodies from a horse. If you remember, we were talking about why it was the horse. That was also passive antibody transfer. You're just giving somebody some antibodies from a tube. And in all of those cases, one thing that we should always remember is that here we're transferring antibodies, and antibodies are just a protein. That means they have a half-life, just like every other protein. They can get degraded. We're not, in this case, transferring a cell that makes the antibodies. We're just transferring the antibody proteins. So eventually in this baby, these antibodies that came from mom are going to get degraded and go away, and the baby will be fine. And now it'll be totally fine because we know some other treatments. But so with any of these kind of passive antibody situations, because we're transferring just the antibody protein, the pre-made protein, not the cell that's actually doing the production, it's only something that's going to be happening for a short time. And you can see this here, the antibodies that are coming from mom go away because we're just transferring those proteins and not the actual cells that produce the antibodies. Mentioning the cells here is important because now I'm going to switch gears into thinking about the issue of diversity of antibodies and B cells. I'm going to actually do the next couple slides in a different order. See if maybe this makes some sense. This is always a place where like either you really get it or you really don't. So I'm gonna try something slightly different here. So one thing I want to tell you is that the goal of the next bit of time is that we're going to be thinking about how we get diverse antibodies. How we basically generate antibodies in the body. Both today and Wednesday, we're going to be talking about this in really kind of genetics and molecular biology details. We're going to be thinking about the DNA and the enzymes and stuff. And then next week, we're going to be thinking about this more in this context of a cell. And we're going to think about like how the cell controls all of this business. But what I want you to realize is that the DNA and enzymes and stuff that we talk about today they're all in a cell. Okay? So we're going to be looking at B cells. We're going to be thinking about B cells potentially making either a B cell receptor or an antibody. And the antibody is actually just a secreted version of that B cell receptor. So if I happen to talk about the B cell receptor or the antibody interchangeably, that's because, in fact, when we're generating this diversity, when we do the work of generating the diversity, that gives us both a B cell receptor and an antibody. So just know that if I use one term interchangeably, it's because they're the same thing. There were lots of observations about B cells that were made before the actual process, before the answer was discovered. So I want to tell you about a couple of different observations and sort of big picture things we knew here. This slide uses a vocabulary word that students find sometimes a little confusing. I'm just going to give you a broad definition of it now. You are going to see you're going to kind of get it in more detail next week in another way. So know that we're going to define this again. But one of the things that has been observed about B cells and their ability to make antibodies is that they do this process called allelic exclusion. And what you need to know about for allelic exclusion right now is that each individual B cell, and this thing on the bottom is our B cell, makes one antibody / B cell receptor. So here it's showing you a B cell receptor. This same B cell can also make this exact same thing in antibody version. But it can't make a different one. It can make this one that has the blue heavy chain with the diagonally binding part and the red light chain with the diagonally binding part. It can make like 1,000,000 copies of that one, but it can only make that one. Is basically what allelic exclusion.
Not sure I understand.
That's what allelic exclusion basically is telling us. We're getting a B cell with homogeneous receptors and antibodies. So each B cell makes one receptor / antibody type. And it was also observed that there were a couple of other things that went on. One of the things that was observed was that if you saw the same antigen second time, the response improved. And so here we'll look at this darker blue, which is listed as the adaptive response, and today we'll call it antibody responses. And you can see that the antibody response takes a little while to get started, but then it gets pretty big, and it gets really big a second time around. So it can improve over time. So somehow that B cell is somehow like improving over time. And what we realized is that this sort of process was what was happening with B cells. And so this is like a key principle of all of adaptive immunity and all of immunology, which is that we know that there's this theory called the clonal selection theory. And so what we now understand is that at the beginning, we start from a stem cell, and that's a stem cell that is eventually going to grow up to be a B cell. It has decided, I want to be a B cell when I grow up, but it has not yet done anything else. And it's going to go through this diversity process that I'm going to be telling you about next. And it's going to make a bunch of different progeny. So it's going to make progeny number 2 and progeny number 3. Or in this other view from a different book, it's going to make red, yellow, and orange, each with slightly different receptors. And those are all being made in some primary lymphoid organ. You can kind of imagine that they're being made off in some little area where the developing cells are like sequestered. So they're in like cell development school, off away somewhere. And then when they finished developing, they've made all of these diverse kinds of receptors. They get released to the rest of your body. And they go out to the rest of your body and they search for their antigen. And you can imagine that you make one of every possible kind of B cell early in your life, and you have one of every possible kind of B cell, like we'll say by the time you're 1 year old. I made that number up, but we'll go with that. You make one of each, and that one just floats around your body and looks for its antigen. Some of them will go forever, like this one, and never find its antigen. Like right now, you all have a B cell that can respond to some epitope on Ebola. You all have a B cell that makes a receptor and an antibody that responds to Ebola. And it's floating around your body looking for Ebola. And we all hope that it never finds Ebola. And you go through your entire lifespan and it dies sad with you that it never found its match. Right? That's what we hope. But some of them are going to find their match. And when they find their match, they're going to divide and make sort of a clone army of themselves. And so you can see here over on the right, the red one, the orange one, they did not find their match, but the yellow one did, found its match, and now we've made a whole big clone of these yellow cells. This is actually why we like to call this process, or this whole area, adaptive immunity instead of acquired immunity. Acquired immunity makes it sound like you acquired this cell after you got infected. But you didn't acquire it, you had it all along. It just got better by making a clone army of itself. Basically, and you will see, I will do this so many times this semester, it got a signal that said, you're useful. Here's your pat on the head that says you're good. Make more of yourself. There's a lot of pats on the head. Immune cells, very needy, need a lot of pats on the head. So this cell got a sort of, you did a good job, you make more of yourself. This other cell went around the body the whole time, like, where is my thing? And it never found it. So this is kind of what we've realized is how adaptive immunity works. The reason why this is so critical is that one of, I would say probably the biggest misconception people have about the immune system has to do with some aspects of this diversity process, because this diversity process happens in the absence of antigen. The cells are doing this diversity process off sequestered in some primary lymphoid organ. They're not doing it in response to a microbe. You make one of each now, or when you were a baby. And in fact, you keep doing it for the rest of your life. You just keep making all of them, like one of each. And they just float around your body and hope that they find a match. And you hope that they don't find a match. That's how we get this kind of improvement in secondary responses. So all of that's fine. All of that works really well. But we get a little bit of a problem here. And the problem comes here. I told you about Landsteiner's experiments in the early 1900s and how he was able to make animals make antibody responses against these closely related chemical compounds that he had synthesized. And he was able to say, I think I figured out, that you can make an antibody to anything. I'm going to give you some numbers. The numbers, you will actually see in different textbooks them varying a little bit because there are different ways you can get the numbers. I don't care that you know the specific number. The thing I want you to notice when I show you these numbers is what the problem is. I don't care that you know the exact number. And so if it changes on different slides, it's okay. So from a lot of work, people figured out you could make 10 to the 16th different kinds of antibodies. That basically means 10 to the 16th different B cell receptors also. Probably even means 10 to the 16th different B cells based on what I told you about allelic exclusion. Ten to the 16th B cells, not hard. That's fine. I don't care. With the BCRs and antibodies, what kind of biological molecule is an antibody or a BCR? What kind of biological molecule is an antibody? Yeah, proteins. Okay. Where does a protein come from? Okay, so we had to have a gene encoding the mRNA, and then it gets translated into a protein, right? So in order to make a protein, you need a gene. Yeah? Dunaway was here. He'd be very impressed with our sophisticated drawing on the board. So, fine, all good. After this number was sort of calculated, there were some other kind of calculations that were done. And again, I'm summarizing a lot of things. And we learned a really critical thing, which is how many genes are in the genome. And when we learned how many genes in the genome, we found out that the number was about 2.4 times 10 to the fourth, the number of genes you have in your genome. So what's the problem here? Well, so we can make lots of cells, you can make lots of copies. So it's not the cells that is an issue. Yeah, genes. So we make more of these proteins than we even have genes in our whole genome. And like, not a little bit more, a lot more. Like 10 to the 12th more. That's what is that? That's a trillion times as many. And we know that proteins come from genes, so how the heck can we make that happen? And so this was this problem that immunologists were obsessed with for 70 years, was trying to figure out how the heck do we get this many antibody proteins from this many genes? And I'll even tell you, it's a bigger problem than just that 1 trillion times thing, because 1 trillion times works if we say every gene in your genome, all 2.4 times 10 to the fourth, encodes an antibody. But they don't, because some of them encode T cell receptors who have the same problem. And like, there's even, like, I know it's hard for me to admit, non-immunology genes. Like, so we somehow have to get a huge number of proteins off a really, really small number of genes. And we're going to do this early in a developmental process, like right after the cell is formed, before we even let the cell kind of like go out into the body. So what we're going to be talking about today and Wednesday is how we solve this problem. So we're specifically going to think about how to solve the antibody diversity problem. And I've got the antibody diversity problem listed here as well. It's the same problem that we've seen before. The real answer has a few different parts. I know that some of the terminology on this slide may not make a ton of sense right now. But when you come back and are studying for the exam, you are going to find this to be a very useful little outline. So you can see that there are two parts to the answer. One is something called combinatorial diversity, and one is something called junctional diversity. And each one contains two pieces, which are shown here. And so it's really all of these things coming together that allow us to solve this antibody diversity problem. One thing to also note, and this is something that I used to go into more detail with in class, and I sort of have it out for time's sake, but I just have to at least say a little teensy bit of it. Like I said, people were trying to figure this out for 70 years. There were a bunch of different hypotheses that were really bad. And what was one of the things people realized is that the answer had to be something that went with DNA and genes, because it had to be something that could go from one cell to the progeny cell in cell division. And so sometimes people will think about other types of ways you could do this, and all of them have the problem that they don't really go to a progeny cell very well. Only messing with DNA is going to help us make this work. And I will also tell you that we're going to learn about a scientist who actually did some really important experiments to figure this out. He did the experiments I'm going to tell you about were done in 1976. After he did these experiments, he decided that immunology was over and all the hard questions were answered, because he answered it. And he is still an active researcher today, now doing neuroscience research, because he was just like, yeah, I'm done. I figured out all of immunology. I got it. As a result of these experiments. So in order to help us think about combinatorial diversity and to understand combinatorial diversity, I'm going to give you a little analogy. And we're going to imagine, in my analogy, that I have decided this whole teaching thing is not for me anymore. And I have decided instead I am going to open a restaurant. Now, you snicker a little bit when I say that, but the one reason why you should snicker a lot when I say that is because I can't cook. And so let's imagine that I know how to cook 4 things. This is not that much of an understatement. Let's imagine that I know how to cook chicken. I know how to cook carrots. I know how to cook beef. And I know how to cook broccoli. That's it. That's all I got. Okay? My menu has 4 things on it. It's kind of a little bit of a sad menu, right? And let's imagine that I now learn how to cook a 5th thing. And I now learn how to cook spinach. Now I've gone from 4 things to 5 things. That's not that helpful for my menu, right? But I could be clever about my menu. And I could think about making my menu based on combinatorial diversity. And so I could say, I don't have 5 things on my menu. I have 1 thing, which is chicken and carrots. I also have chicken and broccoli. And I have chicken and spinach. And I have beef and carrots. And I have beef and broccoli. And I have beef and spinach. Before I learned how to cook spinach, I had 4 things just that I could do. And if I made combos, I got 4. But now that I learned this 5th thing, spinach, and I started thinking about combinations, I didn't just add 1 thing to my menu, I added 2. Because I'm counting not as an individual thing, I'm counting as a combination for what we call my menu. Similarly, if I learn how to cook corn, suddenly I get 2 more things on my combo menu. And so I only increased by a small number of actual recipes, but because I'm counting combos, not individual recipes, I have way more things on my menu. And you can imagine how I can take a small number of recipes and combine them in different ways and suddenly get a lot of menu items. And this is how a person who cannot cook like me ends up with the restaurant. So does that make sense about how this works and what combinatorial diversity really means? Yeah, Andrew. You, in fact, could combine more than 2 things. Yes, I'm just simplifying for my example, but you could 100% combine more than 2 things. And so one part of combinatorial diversity is something you already learned about. And that is simply that we combine heavy chains and light chains. And that in order to have an antibody bind to antigen, it's about the combination of which heavy chain and which light chain you have. And so we can have a heavy chain gene and a light chain gene, or a set of them, and do combinations, just like I was doing with my menu, and have a relatively small number of genes give us a relatively large number of antibodies, because the antibody counts as the combo, not as each individual thing. And so one part of combinatorial diversity is just we're going to pair together different heavy chains and light chains. And so if I took this same heavy chain and paired it with like a hot pink light chain, it's another antibody. And I didn't necessarily need a whole lot more genetically. When scientists continued to make observations like this, they started to look at the antibody proteins themselves and do sequencing of the antibody proteins. And they found that you could have the same constant region put together with different variable regions. So you could have multiple different variable regions all connected to the same constant region. You could even have the same variable region with different constant regions. And in total, there were like so many variable regions and a small number of constant regions. From this, they came up with another hypothesis. And that hypothesis was that it wasn't just about combining the antibody, the light chain gene and the heavy chain gene, or the light chain and a heavy chain, that maybe when you have the DNA making up just a heavy chain, for example, you had little pieces. It wasn't one big gene, it was multiple little pieces of genes. And each of our different 10 to the 16th antibodies were combinations of these gene pieces that were different. This is known as the mini gene hypothesis. And so the idea was that maybe you would have a bunch of different options of this blue part and a bunch of different options of the red part, and you could combine them in different ways. You could pick one of these blue ones and one of these red ones and make a combination, and that would be how you would encode the antibody. And you could also have a constant region that was separate that you combined with it. So you could mix and match which constant regions went with which variable regions. And so it was the same thing, you're combining 2 different proteins, but you're also combining mini genes in order to make those proteins in the first place. And so this was sort of what the idea was for the mini gene hypothesis and what they kind of thought was going on. This one's only showing it with 4 combos. So this is my chicken, carrots, beef, and broccoli example. But notice once I got past 4 and started adding new things, the numbers went up greatly in terms of combos. It was just easier to draw with 4. So here you can see there are 2 mini gene segments called V1 and V2. And imagine the cell has to pick a V. It could be pick V1 or V2. And it's got a couple options for J1 and J2. And the cell will actually choose two of them and physically cut and paste the DNA to put those two next to each other. So here you can see this cell chose V1 and J2 and pasted them together next to each other and took all the other V's and J's and threw them away. And so that one developing cell made its own unique combo of this part of the chromosome, of this DNA. And so this developing cell picked V1 and J1. See, it's red and it's yellow. And this one picked V2 and J2. And this one picked that one was V1 and J2. This is V2 J1, V2 J2. And so with a relatively, not even full genes, little gene pieces, but we make combos of them, we can suddenly get to a bunch of different receptor combinations really quickly with a very small amount of genetic information. So this was kind of what the hypothesis suggested. The next slide has an experiment, and I'm going to tell you sort of key points of the experiment. Some of you who have a lot of molecular biology background may say, I want to know more details of it because I'm skipping and only hitting the highlights here. I'm happy to go over as much detail as you want in office hours. But this is one of the experiments that was done by this scientist, Tonegawa. And Tonegawa took two different kinds of cells. One of those sets of cells were cells that made antibodies, which are shown in green. And one of those cells were here listed as liver cells. He actually did a bunch of different types of things here. The point of these cells are that they are cells that shouldn't have done this magic DNA stuff. They are cells that are not immune cells, not making antibodies, should not have messed with the antibody DNA. Because his hypothesis is that developing B cells mess with their DNA compared to the rest of the organism. So actually in some of his papers, you'll find this was actually sperm, because he's like, well, sperm definitely shouldn't be messed with. Some of them will be liver cells. It doesn't really matter. It's antibody producing cell, matching cell that doesn't produce antibodies and thus shouldn't have messed with this DNA. And he took the DNA out of both sets of cells. And he ran that DNA on a gel. And what you can see is that he got basically all the sizes. It was like a total smear of DNA. Because, of course, these cells, no matter what kind of cell they were, had a ton of DNA in it. Right? But he didn't care about like every single bit of DNA in those cells. He cared specifically about the gene encoding the potential antibody. He cared about kind of this part of the DNA, the part with these little mini genes. He was hypothesizing that in the cell that was not an immune cell, that was not making antibodies, all of these little pieces would be separate from each other. And he hypothesized that in the cell that was making the antibody, the pieces would be together. He made a radioactive probe that could hybridize to the DNA that contained V1 and J2. I don't know what numbers V and J they were, but we'll call them V1 and J2 for our purposes today. So he made radioactive DNA that could hybridize to that. And then he looked at the DNA that he ran. Now, if you look at this configuration from the cell that's not an immune cell, and you think about the radioactive probe that hybridizes to V1 and J2, how many things is it going to hybridize to? How much hybrid is that? What are you going to see? Having not known this experiment, that question may not make a ton of sense. You're probably going to see hybridization to two places, right? The V place and the J place, which are not next to each other. And when you hybridize here, you're probably going to see hybridization to one place because that DNA moved and was next to each other. The DNA, in fact, is going to probably be a different size when you get the hybridization because the DNA has been messed up between the antibody producing cell and the original germline cell. And so that's actually what he saw. He saw that when he looked for the probes that bound to the V and the J segments in the germline DNA, in the liver where the DNA wasn't rearranged, he got two bands. The DNA, the thing that was hybridized to two places. And when he looked at the antibody producing cells, he saw one band. The DNA was now only in one place. The DNA had moved together. And so he realized, like, yeah, this hypothesis that this DNA is being cut and pasted like this is correct. He also had one other prediction that came out of this hypothesis, this mini gene hypothesis, and I put up this image on purpose. What is one other thing you might imagine, you might hypothesize he could find if this process of lining up the DNA, cutting out the intervening sequence and making this little circle and pasting this DNA back together? What might he be able to find if he looks real closely? Yeah, Emma. So he might be able to find some of the specific sequence, and that kind of is what he did with that, with the experiment I just showed you. But there's one other thing he could maybe find if he looks real close. Yeah, Andrew. So he could see that the cell will make the right protein, sure. Yeah, real easy, guys. Yeah. Yeah, he's like, if this was totally true, you should also be able to find just a random circle of DNA chilling in the cell that got thrown out, right? And he did. So he could also easily show those random pieces of DNA. You can see they vary a lot in size and in specific details, but he could find the excised DNA that had been removed in those cells. And he couldn't find it in the cells that were not making antibody. And so, like I said, got a Nobel Prize. He said, immunology's over. He's like, we're done. So this is now officially how we know that our B cells work in order to make antibody genes. This happens in an antibody-producing cell twice. Once to make the heavy chain, and once to make the light chain. With the heavy chain, we have 3 different mini gene segments. So Andrew wanted to know if we could have more than 2 in my recipes. We totally can. In fact, the heavy chain has more than 2. It has one called the V segment. Originally, it was called the variable segment. You don't care. It has one called the D segment. Originally, the diversity segment. Still don't care. And the J segment, the joining region. And each B cell, in order to make a heavy chain, chooses a V, a D, and a J and pairs them together upstream of a constant region. And that constant region might be C mu if we're making IgM, or it might be C delta if we're making IgD, or gamma if we're making G, epsilon if we're making E, alpha if we're making A. You can see that if we actually think about the antibody protein itself, the V segment that was chosen by the B cell is actually encoding the first 101 amino acids of the antibody protein. The D segment is encoding amino acids about 102 to 106. The J is about 107 to 123. And that B cell will also do this process again to make a light chain. It's a different chromosome, a different region of the genome. And it will combine, in this case, just a V segment and a J segment with some kind of constant region. And the constant regions for the light chain are different than the constant regions for the heavy chain. In the case of the light chain, we have segments 1 through 97 encoded by the light chain V, and about 98 to 110 encoded by the light chain J. So I'm going to make a little bit of a drawing on the board right now. Part of the reason why I'm doing this drawing is because in the problem set that you're going to have for this, there are some places where you're going to have to make some drawings. Today's drawing is not complete. There are other pieces that need to go into the drawing that will happen next time, but this is going to start you thinking about how I imagine some of these drawings. So if I was going to draw a heavy chain at the start, I might draw a V1, I might draw V2, I might draw V3, then I might draw D1, D2, D3, J1, J2, J3, and then my 5 different constant region choices. MDs give everyone apples. So that might be what a heavy chain will look like, not complete because there's something that's not there. And a light chain might look something like this, because I haven't told you the name of the constant region yet. And so this is actually what it really does look like. The heavy chain is encoded on chromosome 14. And there are about 40 V regions, about 23 Ds, and about 6 Js. And then actually we have 2 different light chain regions in 2 different chromosomes. They're called kappa and lambda. That's why I just wrote constant there, because technically it's either kappa or lambda. And you can see that there are 30 Vs and 4 Js, or 35 Vs and 5 Js in the light chain. I'm going to skip over the next slide and deal with that next time, just to give you an idea. So the cell separately does this process. We often refer to this process as VDJ recombination. Recombination is the term for cutting and pasting DNA to make new combinations. And we are combining a V, a D, and a J in the case of heavy chain. We just call the whole thing VDJ recombination as the process. And so you can see each developing B cell does VDJ recombination twice, one to make a heavy chain, one to make a light chain, pairs those 2 different things that's made together, and that's how we get to that number that is so impressive. And so this kind of gets us to that same area. So with the heavy chain, we've got 45 Vs, 23 Ds, 6 Js. So if we multiply 45 times 23 times 6, we have 6,210 different heavy chains we can make out of less than 100 genes. And then if we look at the light chain, we end up being able to make either 205 or 165, again, with a relatively small number of different genes. And if we take those 2 things, all the different heavy chains and multiply by all the different light chains because of all the different combos, we get up to 2 million different antibody proteins we can make out of less than 200 genes. So 2 million proteins out of 200 genes. Pretty good ways of sort of dealing with genetics here. We're going to talk a little bit more about some of the specific details of what we should be looking for with VDJ recombination. I may, now that I'm looking at it, post a second set of slides just where I drag some of these in a different order for Wednesday, but I'll see you guys Wednesday. Wednesday, we're going to be in the nitty gritty of enzymes and DNA, and I'm going to be drawing sequence on the board. So be ready for your molecular biology on Wednesday.